to talk about the electron transport chain. A summary of this is basically it's a series of protein complexes that are embedded in the inner mitochondrial membrane, and electrons are captured from donor molecules, usually NADH, and these electrons are transferred through these protein complexes. And coupled with the transfer of those electrons is the pumping of hydrogen ions, or protons, across the inner mitochondrial membrane. This proton gradient is then used by ATP synthase to make ATP. So now we're going to look at each of the different proteins and what they do. The first protein is NADH dehydrogenase, and that's here. And it takes 2 electrons from NADH, or whatever the donor molecule is. Next, it's going to pump 2 protons across the mitochondrial membrane. So we're going to take in 2 electrons and 2 protons. And it's going to spit those 2 protons out up here. And then we'll talk about the electrons here in a second with our second protein, which is more of a transfer protein, and that's ubiquinone. And that's here. Ubiquinone has the job of transferring those 2 electrons from NADH dehydrogenase to cytochrome BC1. So this is ubiquinone here, and we're going to show it's moving our 2 electrons. Next we have cytochrome BC1. Cytochrome BC1 is going to pump a proton across the membrane for each electron given to cytochrome C. And cytochrome BC1 we have represented here. So it's going to take 1 electron at a time and give it to cytochrome C. Cytochrome C is going to transfer electrons individually from cytochrome BC1 to cytochrome oxidase. And cytochrome oxidase, let's label this one here. For cytochrome C we had right here. Cytochrome oxidase we have here. And cytochrome oxidase is going to react 4 electrons. So once it gets 4 electrons from cytochrome C, it's going to do this. It's going to react 4 electrons, 1 oxygen molecule, and 8 protons to form two water molecules. And if you do the chemistry there, you'll see we still have 4 extra protons. What it does is it pumps these 4 extra protons across the membrane. And this has all been leading up to just setting up this proton gradient at this point. So all those protein complexes do is set up that gradient, and they're setting that up for ATP synthase. ATP synthase is our last big molecule there. So we're going to kind of label this to the side, because as you can see, there isn't a whole lot of room on it, because it's pretty complex. So ATP synthase uses that proton gradient to spin the molecule like a turbine, essentially. This is going to alter the molecule shape. Because as you can look at the ATP synthase molecule, you can see that top portion and bottom portion are kind of connected by a shaft. So when the top portion spins, that shaft spins the bottom portion and will change that shape. And that change of shape facilitates the bonding of ATP, the bonding to form ATP from ADP and an organic phosphate. To form ATP. And whenever this happens, it happens with the transfer of 3 protons across that membrane. And that transfer is going to be across or down its gradient now. So we're releasing that potential energy to help us bond. So looking at this process, we create a lot of ATP using those NADHs that we produce a lot of times during our other cell respiration processes. So the majority of ATP in cellular respiration is made through the electron transport chain. And that is all about our electron transport chain.